Dear students, a warm welcome to everyone. We all accept rice as a part of our daily meal, but we are not taking rice alone as a food. Pizza is the favorite food for everyone, but we are not advised to consume it regularly. There are some cheese lovers who taste all types of cheese. These cheese provide more calories than any food, but neither pizza nor cheese is preferred choice of our regular meal. We South Indians prefer a complete meal with rice, dal, rasam, curd and some other extra items to satisfy our hunger and energy requirements. The Food Council recommends and groups the food items to be eaten most, lot, moderately and least. So, what is the main reason behind the classification? Yes, it is to provide ourselves with proportionate quantity of carbohydrates, protein, fat, vitamins and minerals to regulate all the metabolic activities. This proportionate quantity provides us the balanced diet. When you consume a normal meal, you will get energized and do your work as per the schedule. But if you are provided with a hot biryani, then after few minutes you will feel drowsy. So what is happening in your body after biryani is tasted? Whether it is pizza, cheese, full meals or biryani, they will supply the essential nutrients for the growth of cells and tissues by regulating DNA replication, transcription and translation. The so-called central dogma of the cell, which instructs the cell to provide the functional products, the so-called enzymes and proteins. Basically, the food we eat gets converted into simpler molecules and ultimately provides energy to the cells to regulate its key metabolic functions. A major challenge in bioprocess development is to select an organism that efficiently make a given product from a particular substrate. The metabolism of substrate is the key in realizing the passage of cellular information. With the advent of genetic engineering, it is possible to remove and add genes to an organism to alter its metabolic functions in a predetermined way. It is essential for the bioprocess engineer to understand the key factors of major metabolic pathways and products of various cell types. In today's class, we will be studying the impact of key nutrients in regulating the cell's metabolic activities as living cells require energy for biosynthesis, transport of nutrients, motility and maintenance. This energy is obtained from the catabolism of carbon compounds, mainly carbohydrates. These carbohydrates are synthesized from carbon dioxide and water in the presence of light by photosynthesis. The metabolic reactions are fairly complicated and vary from one organism to another. A major challenge in bioprocess development is to select an organism that can efficiently make a given product from a particular substrate, and it is also a tedious task to identify the suitable substrate that could be utilized by the organism and produce metabolite. Differences in microbial metabolism can be attributed partly to genetic differences and or to differences in their responses to changes in their environment. Even the same species may produce different products when grown under different nutritional and environmental conditions. Even the same species may produce different products when grown under different nutritional and environmental conditions. The control of metabolic pathways by nutritional and environmental regulation has become an important consideration in bioprocess engineering. For example, the Saccharomyces cerevisiae, commonly known as baker's yeast, produces ethanol when grown under anaerobic conditions. However, the major product is yeast shells, when growth conditions are aerobic. Even under aerobic conditions, at high glucose concentrations, some ethanol formation is observed, which indicates the metabolic regulation not only by oxygen but also by glucose. This effect is known as Crabtree effect. The ethanol formation during Baker's yeast fermentation may be reduced or eliminated by culture with intermittent addition of glucose or by using carbon sources other than sucrose and glucose that support less rapid growth. Here is the nutshell of all the pathways that happens in the cell when glucose is used as a carbon source, wherein the key products that are formed upon utilization of glucose are glycogen, G6P, pyruvate, acetyl-CoA, and so on, wherein we are able to produce a huge number of products that are required for regulating various metabolic activities in the cell. As a result of various catabolic and anabolic reactions occurring in the cell, we are able to produce the amino acids and nucleotides that are responsible for formation of proteins, DNA and RNA to instruct the cell's internal machinery to regulate its activities. The metabolic reactions are fairly complicated and vary from one organism to another, but the major categories of metabolic reactions are: first comes the fueling reactions, which aids in degradation of nutrients. Second, the biosynthesis of small molecules, where the amino acids and nucleotides are produced. Third comes the polymerization reactions, which aids in biosynthesis of large molecules. And finally, the assembling reactions, where the smaller molecules will be assembled to larger entities. These reactions take place in the cell simultaneously, and as a result of metabolic reactions, end products are formed and released from the cells. These end products of each reaction are highly valuable products for human and animal consumption. The schematic diagram of the reactions and processes involved in cell growth and metabolism and product formation is given here. Substrates are taken up by the cells by transport process and converted into precursor metabolites via the fueling reactions, and the precursor metabolites, including the nucleotides, amino acids, hexamides, fatty acids and sugars, are converted to building blocks that are polymerized to macromolecules viz. DNA, RNA, proteins, peptidoglycan, lipopolysaccharide, lipids and glycogen. Finally, the macromolecules are assembled into cellular structures like membranes, organelles, envelope and so on that make up the functioning cell. Precursor metabolites and building blocks may be secreted to the extracellular medium as metabolites, or they may serve as precursors for metabolites that are secreted. The cell may also secrete certain macromolecules, primarily proteins, that can act as hydrolytic enzymes, but some cells may also secrete polysaccharides. The energy in biological systems is stored primarily and transferred via ATP, which contains high energy phosphate bonds. The active form of ATP is complex with Mg2+. The standard free energy charge for the hydrolysis of ATP is 30.5 kJ per mole. The actual free energy release in the cell may be substantially higher because the concentration of ATP is often much greater than that for ATP. Biological energy is stored in ATP by reversing this reaction to form ATP from ADP and phosphoric acid. Similarly, ADP dissociates to release energy. The structure of ATP is given here, which is a nucleotide base with 3 phosphate molecules and undergoes hydrolysis to form ADP, and this ADP, when reacted with the phosphoric acid, in turn forms the ATP, and The analogic compounds of ATP is the GTP, uridine triphosphate and cysteine triphosphate. These compounds also store and transfer high energy phosphate bonds but not to the extent of ATP. High energy phosphate bonds are produced during metabolism and each energy in ATP is transferred to lower energy phosphate compounds such as glucose 6-phosphate and glycerol 3-phosphate. Biological oxidation reduction reactions are carried out by nucleotide derivatives, especially the NAD plus molecules, which are nothing but the nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. The oxidation reduction reactions are readily reversible. NADH can donate electrons to certain compounds and accept from others depending on the oxidation reduction potential of the compounds. NADH has two major functions in biological system. First comes the reducing power, in which the NADH and NADPH supply hydrogen in biosynthetic reactions such as carbon dioxide fixation by autotrophic organisms. The ATP formation in respiratory metabolisms is also guided by these NADH molecules. The electrons carried out by NADH are transferred to oxygen via a series of intermediate compounds. The energy released from this electron transport results in the formation of up to 3 ATP molecules, and ATP can be formed from the reducing power in NADH in the absence of oxygen if the alternative electron acceptor such as nitrate ions is available. The oxidation reduction coenzyme nicotinamide is given here, where it can accept the hydrogen ions to form the active molecules. The aerobic catabolism of organic compounds such as glucose may be studied in three different phases. First, glycolysis or Embden-Meyerhof pathway for fermentation of glucose to pyruvate occurs. Second, the Krebs or tricarboxylic acid or citric acid cycle for conversion of pyruvate to CO2 and NADH. In the third phase, respiratory or electron transport chain for formation of ATP by transferring electrons from NADH to an electron acceptor occurs. The final phase, respiration, changes reducing power into biologically useful energy form ATP, and the respiration may be aerobic or anaerobic depending on the final electron acceptor. The bioreactions involved in breakdown of glucose by glycolysis is given here, where the glucose undergoes a series of phosphorylation, decarboxylation and hydrolysis reactions to form the end product pyruvate. In the Embden-Meyerhof pathway, the glucose undergoes phosphorylation and decarboxylation reactions to form ribulose 5-phosphate, and this ribulose 5-phosphate undergoes a series of reactions to form sidoheptulose 3-phosphate and erythrose 4-phosphate, and these molecules will undergo further biochemical reactions to produce pyruvate. Shortly, the glucose that is fed into the reactor as the substrate for the growth of organisms is subjected to glycolysis for the production of ATP and NADH molecules. The ATP is the energy precursor of the cell. Pyruvate is the end product of glycolytic pathway. This pyruvate enters into the mitochondria of the cell to form the ATP in the presence of oxygen, and in the absence of oxygen, this pyruvate is fermented to produce the lactate and carbon dioxide. In the mitochondria, the TCA cycle is initiated, which aids in production of various other biomolecules that are required for production of energy. The overall stoichiometry of glycolytic pathway is that glucose in the presence of 2-ADP, 2-phosphoric acid and 2-NAD plus molecules produces 2-pyruvate, 2-ATP, 2-water and 2-NADH and 2H plus ions. The fate of pyruvate molecule is decided by the presence of oxygen in the reactor. If in the presence of oxygen, it enters the mitochondria, goes for TCA cycle, and in the absence of oxygen, it undergoes fermentation process to produce lactic acid. So, a glucose molecule can be grouped up to form 12 NADPH and 6 carbon dioxide through 6 passages in the loop via G6P, PP pathway, F6P and G6P. The key factor to be understood here is that NADH is produced together with NADPH in catabolic reactions, but in the anabolic reactions, the NADPH is consumed. The net ATP yield in glycolysis is 2 mole of ATP per glucose under anaerobic conditions, and acetyl-CoA is transferred to the mitochondrial membrane at the expense of conversion of 2-NADH produced in glycolysis to FAFDH2, and the acetyl-CoA is a key intermediate in the metabolism of amino acids and fatty acids. The key scheme of pyruvate metabolism is given here. The pyruvate in the presence of pyruvate dehydrogenase forms acetyl-CoA, and this acetyl-CoA actually enters into the TCA cycle, also known as Krebs cycle, consists of series of enzyme catalyzed reactions that is of central importance in all living cells that use oxygen as part of cellular respiration. In eukaryotes, TCA occurs in the matrix of mitochondria. In aerobic organisms, the Krebs cycle is a part of metabolic pathways involved in the chemical conversion of carbohydrates, fats and proteins into carbon dioxide and water to generate a form of usable energy. Other relevant reactions in the pathway include those in the glycolysis and pyruvate oxidation. In addition, it provides precursors for many compounds including some amino acids. This cycle is called as TCA because of production of tricarboxylic acids such as citric acid, succinic acid and isocitric acid. Here, the condensation of acetyl-CoA into oxaloacetic acid results in citric acid, which is further converted to isocitric acid and then to alpha-ketoglutric acid with a release of carbon dioxide, and further succinic acid is produced, which is further oxidized to fumaric acid. Then dehydration of fumaric acid to malic acid and oxidation of malic acid to dehydration of fumaric acid to malic acid and oxidation of malic acid to oxaloacetic are the 2 last steps of TCA cycle, and for each pyruvate molecule entering this cycle, 3 carbon dioxide and 4 NADH plus H plus and FADH2 are produced. The overall reaction of TCA cycle is given here, where the reducing powers produced is used either for biosynthetic pathways or for the ATP generation through the electron transport chain. The major roles of TCA cycle is to provide the electrons for the electron transport chain and biosynthesis, to supply carbon skeletons for amino acid synthesis and to generate energy. The next key important topic in bioenergetics is the respiration and controlled sites of aerobic glucose metabolism. The respiration reaction sequence is also known as electron transport chain. The process of forming ATP from the electron transport chain is known as oxidative phosphorylation. The electrons carried by NADH, H plus and FADH2 are transferred to oxygen via a series of electron carriers and the ATPs are formed. 3 ATPs are formed from each NADH plus H plus and 2 ATPs are formed from each FADH2 in eukaryotes. The major role of electron transport chain is to generate NADs for glycolysis and ATPs for biosynthesis. The term the oxidation of NADH and flow of electrons to the electron transport system leading to the transfer of proton from the inside and outside of the membrane is given here. The tendency of protons to return to the inside is called as proton motive force. The ATP synthesis occurs as protons. The ATPase enzyme uses the proton motive force for the synthesis of ATP. The cytochromes, cytochrome A and cytochrome B, and the coenzyme ubiquinone are positioned at or near to the cytoplasmic membrane, and when electrons pass to the respiratory chain, protons are pumped across the membrane. When the protons re-enter the cell through the action of the enzyme FO, F and ATPase, ADP may be phosphorylated to form ATP, and the respiratory chain is therefore referred to as oxidative phosphorylation. The number of sites where the protons are pumped across the membrane in the respiratory chain depends on the organism. In many organisms, there are 3 sites, and ideally 3 moles of ATP can be formed by the oxidation of NADH, and FADH2 enters the respiratory chain at coenzyme ubiquinone. The electrons therefore do not pass the NADH dehydrogenase, and the oxidation of FADH2 therefore results only in pumping of protons across the membrane at 2 sites. The number of moles of ATP formed for each oxygen atom used in the oxidative phosphorylation is normally referred to as P/O ratio, and the value of the stoichiometric coefficient indicates the overall thermodynamic efficiency of the process. If NADH was the only coenzyme formed in the catabolic reactions, the theoretical P/O ratio would be exactly 3. But since some FADH2 is also formed, the P/O ratio is always less than 3. Further, the proton and electrochemical gradient are also used for solute transport, and overall stoichiometry in the process is therefore substantially smaller than the upper value of 3. And as an end result of glucose metabolism from glycolysis to NADH and to ATP molecules are formed, and in the oxidative decarboxylation of pyruvate 2 NADH is formed, and at the end of TCA cycle 6 NADH, 2 FADH2 and 2 ATP molecules are formed. As the end result of aerobic catabolism of glucose up to TCA cycle 10 NADH, 2 FADH2 and 4 ATP molecules are produced. The overall stoichiometry in contact with the production of NAD plus and ATP molecules. The glucose that is fed into the reactor utilizes 36 phosphoric acid and 36 ADP molecules. In the presence of oxygen, produces 6 carbon dioxide, 6 water and 36 ATP molecules. The major control sites in aerobic glucose metabolism is the feedback inhibition. Several enzymes involved in glycolysis are regulated by feedback inhibition. The major control site in glycolysis is the phosphorylation of fructose 6-phosphate by phosphofructokinase. The enzyme phosphofructokinase is an allosteric enzyme activated by ADP and phosphoric acid but inactivated by ATP. At high ATP-ADP ratios, this enzyme is inactivated, resulting in a reduced rate of glycolysis and reduced ATP synthesis. The concentration of dissolved oxygen or oxygen partial pressure has a regulatory effect on the rate of glycolysis known as Pascher effect. The rate of glycolysis under anaerobic conditions is higher than that under aerobic conditions in the presence of ATP. In the presence of oxygen, ATP yield is high since the Krebs cycle and electron transport chain are operating. As a result of high levels of ATP, ADP and phosphoric acid becomes limiting and phosphofructokinase becomes inhibited. Also, some enzymes of glycolysis with thiol groups are inhibited by high levels of oxygen. A high NADH and NAD plus ratios also reduces the rate of glycolysis, and certain enzymes of the Krebs cycle are also regulated by feedback inhibition. Pyruvate dehydrogenase is inhibited by ATP, NADH and acetyl-CoA and activated by ADP, AMP and NAD+. Similarly, the citrate synthase is inactivated by ATP and activated by ADP and AMP. Succinyl-CoA synthase is inhibited by NAD+. In general, a high ATP bar ADP and NADH bar NAD+ ratios reduce the processing rate of TCA cycle. The interrelationships of major metabolic pathways in E. coli is given here, where the glucose is subjected to glycolysis, TCA cycle and electron transport chain to produce ATP. The path of glucose catabolism can be tailored by using suitable inducers. In the E. coli cell, glucose enters the cell via glycolysis, EMP and PP pathway and connects to the TCA cycle, and there is no net loss of strength of the fluxes, and the nonlinear functions of concentrations of glucose plays a major role in glucose metabolism. Fluxes are nearly independent of the substrate concentration, whereas in the mammalian cells, glucose and glutamine are major carbon and energy sources. The glutamine typically provides most of the energy required by the cell to respiration. The cultured cells, tumor cells and proliferating cells exhibit high rates of aerobic glycolysis in contrast to normal mammalian cells, and high rates of lactate production similar to normal cells occurs under oxygen limitation, as oxidative phosphorylation is restricted at low oxygen concentrations. Pyruvate is converted to lactic acid as a means of regenerating NAD+ from the NADH generated by glycolysis. To summarize, you have learned some of the elementary concepts of how cells control their composition in response to the substrate. The cells has the ability to sense the availability of substrate due to the transport of substrate from the medium into cytoplasm. The cellular metabolism starts with the transport of substrates into the cell and is concerned with two primary functions, catabolism and anabolism. Catabolism involves the degradation of a substrate to more highly simple products, where anabolism results in the building up of molecules. The bioprocess engineers should be having enough knowledge on the biosynthesis of nutrients, the path of transport of nutrients into cell, and the mechanism of maintenance of nutrients by the cell. Also, the end result of each metabolic reactions of any substrate in a given cell should be known, and the inducers and inhibitors should be used accordingly in order to change the flux and provide a thermodynamic efficiency from the given process. The reference books for today's session are Bioprocess Engineering, Kinetics, Sustainability and Reactor Design by Cegilio and Gleninger's Principles of Biochemistry. Thank you everyone for joining the class.